Good evening, ma'am.
Good evening.
Good evening.
Good evening, sir.
Good evening, Ajay. Shall we start now?
Sir, you can start.
Yes, ma'am. Thank you. Good evening, my dear students. Once again, you all are welcome to the biological session. Last class, what we discussed already, you know about biotechnology. From this part, there are so many basic terms that studied. Now, we start from the polymerase chain reaction, simply called as PCR. So what is meant by polymerase? Polymerase is nothing but the enzyme used for the otherwise called as DNA polymerase, used for the chain reaction. Oh, number of copy will be obtained by this technique. This is very, very important. Suppose if you want to prepare a particular DNA sequence into millions or thousands copies, okay, we can make it in a laboratory. See this one. So this is already we discussed the DNA as a double standard genetic material. The genetic material, it has a two complementary gene each other connected with the hydrogen bond, isn't it? when it undergoes three processes, what are the three processes have been reported here? Denaturation, annealing, and extension. The extension denoted, these are the cycles. So each one is considered as a cycle. First cycle, what will be the end product? So this is the single copy of the DNA molecule, isn't it? After the first cycles, the end product will be 2 copies. This is the second cycle. See that one? The 2 converted into 4 copies. Third cycle, the 4 copy converted into 8 copies. So here given as a 9th cycle, how many copies is there? You have to consider as a 2 power n. So n it denoted as 9 means you will calculate 9 into 9. So 81 copies of the DNA molecule you will obtain at the end of the 9th cycle. So this is the reaction. First, denaturation takes place in this process. Okay, denature means so completely removal of the 2 band separately. How it will be possible? Due to the heating process. When you are giving the heat up to the particular temperature, so what happened? The DNA molecule connected with the hydrogen bond, it undergoes the weak. As a result, the 2 stand completely separated. After that, what happened? You have to add the prop, otherwise called as primers, DNA primer or RNA primer. Maximum they were used RNA primers, a small initiator of the polymerase reactions. This is used for the initiation process. Okay, so the RNA primer is also used. It is nothing but a small nucleotide fragments attached to the DNA molecule. So the other enzymes also involved in it. See that one, the polymerase enzyme. So as a result, what happened? The 2 copy of the DNA stand along with the RNA prop, otherwise called as RNA primers, it will lead to the process of the reactions. So as a result, end of the particular times, what happened? You will get it the 2 stand. This is the process called as extension process. This process called as annealing. After that, annealing means what is that? Cooling process. Denaturation by heat, annealing by cooling. So after that, the leading is also takes place. That is called as extensions. These 3 reactions occurs in the cycles, first cycle. So end of the reaction, end of the first cycle, you will obtain the 2 copy of the DNA molecule. Again, the process will be repeated again and again. How many times you cannot be able to calculate? How many times you need? You can easily. What will happen? You can do it. Okay. So final, you will get the what you need. That copy you will obtain from this technique. So such type of technique is called polymerase chain reaction. Okay, see that one. So this is also another one. Already we saw it. And then next one is recombinant DNA. So what is the meaning of recombinant DNA, my dear? Recombinant DNA is simply called as rDNA. The small r denoted as the recombinant, the first letter of the recombinant. The hybrid DNA produced by joining pieces of the DNA at a specific point, that is called as recombinant DNA. What is the meaning of recombinant? Means the hybrid DNA. What is the meaning of hybrid DNA? You can choose any 2 strain or any 2 plant or any 2 animals. The DNA molecule united by the technique. So after that, you will get the single strand, isn't it? Single strand structure of the both the genes present in it. That DNA is called as, what is that? Recombinant DNA. Welcome, Dhanalakshmi. Okay. See that part.
Yes, sir. Good evening, sir.
Good evening, Pa. Okay. Next one is restriction enzymes. Already been over, but there are so many enzymes involved in, particularly the restriction enzymes. What is the use of restriction enzyme? The restriction enzyme, we can make a cut any location, any origin of the DNA molecules. So these are the basic term from biotechnology. So already studied these things also. Restriction is the enzymes that cut out a piece of DNA at a specific location. That is very, very much recognizing site. Recognize site. Which site if you want to cut? Which one is the desired quality or characteristics, whatever it may be. Okay, we can use this enzyme. We can easily make a cut, and finally we get the piece. So already you know that after getting the piece, it will enter or running into the GL electrophoresis. Finally, you have to find out the sequences. What are the sequences is there? Otherwise called as nitrogen bases is there. Entire sequences we can easily isolated or identified. Okay, which is essential for the what is a DNA fingerprinting technique. Okay, this is very, very important. Next one is stem cell. What is the meaning of stem cell? A precursor cell that undergoes the division and gives rise to a different lineages of differentiated cells. What is the main uses of stem cell? Stem cell mostly used for the what is that? Transplantation, organ plantation, isn't it? That cases or any other accident cases, they were used the stem cell. The stem cell is also isolated from where, you know, during the pregnancy. It is nothing but the mother cell, otherwise called as precursor cell. Precursor means it has the ability, potentiality of to produce the entire organ. Suppose we are taking a kidney cells. Okay. Suppose we want to grow any part of the kidney. Okay. They can place it. Even taken examples, osteoclast. Osteoclast is nothing but the bone cells. Stem cells also used for the bone making process. Okay. These are all are comes under the heading is known as stem cells. Okay. So okay. Go to the next one is sticky end. Where there is a sticky end already we studied in DNA has the sticky end. See that sticky end, a single stranded complementary nucleotides on the end of the double stranded DNA molecule. This is called as what is a sticky end. Single stranded complementary nucleotide on the end of the double stranded DNA molecule. What is the meaning of this one, my dear? Do you understand this part? See that one? So I will show you. So it is better for your understanding. So this is considered as a DNA molecule. Okay. This is considered as a DNA molecule. This is the double stranded, isn't it? DS simply denoted as double stranded. Okay. DNA molecule. Okay. This is the double stranded DNA molecule. Okay. This is the end. This is the end of the double stranded. This is the end, isn't it? This is the end part. From this end part, there is a small, what is that? Extension of the double stranded DNA molecule. This is called as what is that? Sticky end. This is called as what? So this portion is called as what is that? So this portion completely in this area, no? So this portion is called as what is that? Sticky end. Okay. You got it, my dear? Double stranded DNA molecule. It has the extension of the any one of the sides. You can take an example, so whether it may be 5' end, and also another one is called as 3'. Okay, take an example, 5' to 3'. Here you know about the opposite. There is a complementary genus 3'. Here the 5'. So in this portion, complementary to the DNA molecule, but this copy, the fragment is called as what is a sticky end. Okay, so this is the alpha. Okay. Sticky end. Okay. And next one is the very, very important one of the very, very important enzymes. Taq polymerase. Okay, here the spelling mistake is that Taq polymerase. What is the meaning of Taq polymerase? See here, this is indicated as a one of the bacterium. Say their name has given as a Thermis aquaticus. So simply the first letters. See that the first letter of the genus name and two letters from the species names. Taq, T-A-Q. Taq polymerase means what is that? Already you know about the enzymes. These enzymes isolated from the what is that? One of the bacterium is called as Thermis aquaticus. So a heat stable DNA polymerase. Don't forget, my dear. Isolate from a thermophilic bacterium. So the Thermis, it is denoted as the heat related one. Thermis. Okay. You know all about that. Each and every enzyme, it has a specific activity on temperature, isn't it? Analyze me. Do you know the maximum activity of enzymes?
Sir,
Any enzyme, maximum activity of the enzymes. Which temperature will be completely finished, complete their work. Do you know?
no, sir.
Okay. What about that Ajay, my dear?
Sir, is it 30 degrees, sir?
30. What about the room temperature, my dear?
Yes, sir. Sir, like above 40, it stops working, sir. It gets destroyed, sir.
Very good.
So
Exactly.
like from 30 to 40, sir.
No, one more, two more is add from 40.
42, sir.
42 is exactly because the room temperature will be 37 degrees Celsius, isn't it? What about your body temperature? 36.9, that is equal to 37 degrees Celsius. So the room temperature as well as the body temperature are equal. Okay. So when the enzymatic activity, when you are studying the enzymology, the enzymatic activities, the enzyme activity completely up to the level of 42 degrees Celsius. It is normal one. But the Thermis aquaticus, the polymerase enzyme, see that the heat stable, they told, this is very, very important, my dear students. Okay. The heat stable DNA polymerase isolated from the thermophilic bacterium, the thermophilic bacterium, how many temperature it would be like? It will be work. Can you have any guessing? Both Ajay as well as Dhanalakshmi.
So no, sir.
No idea. What about Dhanalakshmi?
No, sir.
Okay. Normally, you know about each and every enzymatic activity will be completed up to the 42 degrees Celsius, isn't it? But in this category, the Taq polymerase, it's one of the heat stable DNA polymerase isolated from the thermophilic bacteria, Thermus aquaticus, and used in PCR. But in this category, 90 degrees Celsius, that enzymes, it will be act. It will act up to 90 degrees Celsius. So the ability it obtained, okay, it can carry the temperature up to the level of 90. Where there is a 42, just imagine, and 90 degrees, where is it? Just imagine. In between, there is a 58 degrees Celsius also possible. So that much of heat stable component, the Taq polymerase. Sometimes they may ask the question, NEET, which enzymes? It is highly stable in heat. Okay, which one of the polymerase enzymes? Like that, they may ask the question. Don't forget, my dear. Taq denoted as temperature, AQ denoted as aquatic. So the name of the microorganism, Thermis aquaticus. Okay, it is highly heat stable DNA molecule, polymerase, DNA polymerase enzyme, isolated from the microorganism. This one, where is the usage? Means PCR is the usage. This is very, very important. Don't forget, my dear. This is also very, very important question. Once again, I repeat for you. Okay. And TI plasma. What is the meaning of TI? Any guessing? Plasmids already you know, isn't it? Plasmids. So you know about Ajay idea? Any idea in this part? TI.
No, sir. No, sir.
What about Dhanalakshmi? Do you have? Because both are very, very important, my dear. That's why I'm asking.
Sir, tumor plasmid.
Very good. One more is you should add. TI means tumor induced plasmid. What is that meaning, Ajay?
So
Tumor
induced
including
plasmid.
Very good, very good. Both of you, please follow it. Tumor inducing plasmid. Okay, the tumor already you know what is by plasmid? An extra chromosomal double standard component present in the bacteria or any other plant cell. It is considered as a plasmid because it has a self-replicating DNA molecules, isn't it? Suppose that extra chromosomal body, that is nothing but the plasmid, found in the Agrobacterium tumefaciens. Don't forget, my dear students. Tumor-inducing plasmid is also isolated from the one of the bacteria is called as what is that? Agrobacterium. Agro means plant. Bacterium, you know, bacterium related one. Agro means it is related to plant or agriculture related. Tumefacian. Tume means you know about tumor. That's why they have given us a tumor-inducing plasmids also isolated from the Agrobacterium tumefaciens. So what will be the effect of this one? You know, these type of plasmid present in the plant or animal, what happen? No, that can produce the ground call disease in plant. What is the meaning of ground call? Ground call is nothing but the one of the dangerous one. Just nothing but the tumor. Tumor. What is the meaning of tumor, my dear? Tumor is nothing but the. Any idea?
Sir, cancer causing cells.
Cancer causing cells, isn't it?
Yes, sir.
Okay. Okay, correct. Cancer causing cells, okay, but it's also multiply into how many times we cannot be able to control. Uncontrolled growth of cells, it forms as a mass is called as what is that? Tumor, isn't it? Whether it may be a different types is that cancer in our board. Okay, but in this case, 3 question is here. TI plasmid isolated from where? Which one of the microorganisms? And then once again, the Agrobacterium tumefaciens, okay, which one of the plasmid we can separate? These two also very responsible for the which type of disease. So 3 questions here. Don't forget, my dear. See, this is the, and the next one is tool of recombinant DNA technology. Suppose if you want to do any work, need some tools. But likewise, the molecular biology, particularly genetic engineering and also DNA technology, they need some tools for the recombinant DNA technology. So that tools are what are they? You know, restriction enzymes already we studied, polymerase enzyme already we studied, ligases already we studied, isn't it? A restriction enzyme, what happened? You can make a cut anywhere. Polymerase multiplication result mostly used for the PCR. Ligases, after making the cut with the help of the restriction enzyme, we can use the ligase enzyme, DNA ligases. These 3 are enzymes, and also they need vectors. Vectors is nothing but the mediator, and also the host organisms. These are the vector is nothing but the plasmids or DNA or RNA. These are the vectors. Host organism means mostly they were used, which one, you know, bacteria, and also another one is called as plasmids and cosmids and bacteriophages. So these are the tools mostly they were used for the recombinant DNA technology. You must note it down. What is meant by recombinant DNA technology? DNA recombinant again combined together as a result form is no new one. 2 species DNA molecule combined together and form a new DNA sequence. So that technology is called as recombinant DNA technology. In this process, we need these are the tools. So restriction enzyme, polymerase enzymes, and the ligases and vectors. The vectors is nothing but the plasmid, cosmid, and bacteria, and also protozoa, and also host organism is also same. Okay. And the next one is the adenosine deaminase. It's nothing but one of the enzymes. Okay, adenosine deaminase, simply called as ADA. Okay, sometimes they may ask the question, what is the ADA deficiency? Okay, it is also very important functions of the immune system. See that one, deficiency of ADA enzymes, because deaminase, AAC is a suffix, so enzymes. ADA enzyme is crucial for the immune system to function. This disorder is caused due to the deletion of gene for adenosine deaminase enzymes. Sometimes what happen, you know the very well about the chromosomal aberration, addition, deletion, substitution, inversion, transformation, translocation, all those things already in our lab, isn't it? Here they told it is related to the immune system functions. During that time, what happened, the particular DNA molecule, the particular enzyme production will be completely deleted. So as a result, the lack of this enzyme, it is also related, it is also exposed in the immune system function. Okay, media. So this is also another important things. Don't forget ADA, mostly used for the immune system functions. Particularly, this is caused due to the disorder. Disorder means what is that? Abnormalities, chromosomal abnormalities, DNA abnormalities. Can you imagine? Deletion means completely removal of the particular gene. So that particular gene is responsible for the adenosine deaminase. Don't forget. Okay. Okay. See here. So I will show you. So that one is also for your okay. See that. Okay. See that one. So this is considered different chromosome. This is this is not like that. So this is considered a chromosome, isn't it? Chromosome means you know about it, isn't it? So this is the considered the chromosomes, isn't it? This is the chromosome, isn't it? Chromosome. This chromosome, there is a gene is there. Particularly, this is considered genes, and also the locus. This portion is called as locus. The locus, it contains genes. The gene sequences will be like this, isn't it? Already we saw it in the previous chromosomal abnormalities, isn't it? See that one? So this is considered arms, single arms only I have taken from this one. Okay. So in this category, you know about the different, what is that? Combinations. So for example, A, B, it may be a capital A or capital B and C and D. Likewise, the gene arrangement like this. Just imagine this one, and this one is also. Likewise, its arrangement will be like this. So first you take an example, ADA, isn't it? A, so D, ADA, adenosine deaminase, isn't it? What they told, the ADA is essential for the immune function, isn't it? You can take an example, this portion, okay? Completely removal of this portion, what happened? The ADA synthesis will be completely lack. How the gene sequences is there? Just imagine. So like this, isn't it? So a deletion process completed means the structure will be like this. The structure will be like this. After complete removal of the DNA strand, so this portion completely removed it. So removal of this strand, otherwise called as deletion, isn't it? Deletion. So it's a deletion. What happened? Okay, this portion is deleted. So as a result, what happened? The ADA synthesis will be completely lagged. So remaining, how it will be the molecular basis of the chromosome. See that one. So this one is B and C and D. One A is that. So A is completely removed because the particular gene is responsible for the synthesis of adenosine. So this one is completely removed from this portion. Okay. So the ADA is not synthesized because the ADA synthesis of responsible gene is A. So that's why. So now what happened? The particular enzyme is not synthesized. It's also exposed into the where? In the immunological effect. Okay, see this one. Okay, media. Okay, so next BT cotton. So this is also another important. Okay, BT cotton. So BT cotton is not nothing but the Bacillus thuringiensis. Both of you, my dear, Dhanalakshmi as well as Ajay, please note the microorganism, my dear. This is very, very important. Bacillus thuringiensis cotton. It is a transgenic plant. Don't forget, the BT cotton is nothing but the transgenic. What is the meaning of transgenic? Transformation of gene from one place to another place. Okay, don't forget. Okay, transgenic plant. BT toxin genes were isolated from the Bacillus thuringiensis. Okay, thuringiensis, and were incorporated into the cotton plant. Don't forget. The BT cotton, why it is called as BT cotton, you know? It is one of the transgenic plant. Transgenic means what is that? Isolated of the genetic material from the cotton plant. Okay, and also animal source, the microorganism, Bacillus thuringiensis. Here there is one of the genes isolated. That isolated is incorporated with the cotton plant. Source from the animal to plant. Okay, so it has the BT toxin gene were isolated from the Bacillus thuringiensis and were incorporated into the cotton plant. This is the one of the technique we will study. What is BT cotton? How it will be responsible for the genetic engineering. How, what are the effect it will be like that. Okay, my dear. Go to the next one is CRY genes. CRY, CRY genes. The BT toxins are coded by a gene name is called as CRY gene. Don't forget. BT cotton, Bacillus thuringiensis. Okay, the Bacillus thuringiensis is nothing but a prokaryote, otherwise called as bacteria. Isolating the one of the gene. That gene is incorporated with the cotton plant. Okay, that particular gene is called as CRY gene. Which one is responsible for the BT toxins? See, the toxins are coded by a gene named CRY genes. Don't forget, my dear. Another one is called as CRY protein. Okay, these three questions very, very important, my dear. In biotechnology aspects, compulsory. Already I told you, because BT cotton, tumefacin, TA plasmid, see that one? So tools is not right. So this one, TA plasmid, CAC polymerase. These three is very, very important. So, tumefacin, Thermus aquaticus, max. Sometimes they may ask the question, match the following. Okay, and CRY proteins. This is also very, very important because it's also related to the Bacillus thuringiensis. See, so they both are same. Okay, the insecticidal protein. Don't forget, my dear, insecticidal protein. Which one is this? Following insecticidal properties, like that they may ask the question, CRY gene or CRY proteins. Okay, the CRY protein is which is produced by the soil bacterium named Bacillus thuringiensis is called as CRY protein. So this is also another importance. BT cotton, CRY genes are isolated. Okay, the CRY genes toxic substance is called as BT toxin. Okay, the gene is isolated from the Bacillus thuringiensis, right? Okay, it produces the particular gene is called as what is that? CRY gene. The CRY gene is also produced the one of the protein is called as CRY gene protein, otherwise called as CRY protein. Where? From the Bacillus thuringiensis. So the organism is same. The gene is CRY gene. The protein name is called as CRY protein. So don't forget BT cotton. Example, Bacillus thuringiensis, microorganisms. The gene is responsible for the production of BT toxin, that is called as CRY gene. The protein molecule is called as that CRY protein. Which one? The CRY gene produces CRY protein molecule. Okay, these are the important. And next one is a C peptide. What is the meaning of C peptide? Okay, you know what about that? In mammals, including humans, insulin is synthesized as a pro-hormone. Normally, you know what about all the enzymes or hormones, it will not secreted direct active hormones. Okay, this component sometimes what happen, you know, they may produce in the form of, how it will be, in the prior form, pre-hormone. Okay, pre-hormone stage. Okay. Pre-hormone. The pre-hormone undergo the development or maturation, then only it will act as a matured one. Likewise, the insulin, take an example of, you know what about the insulin? It has a peptide bond. It's also having a 2 alpha and beta. Likewise, which one you know? Hemoglobin. But in this category, A and B chain is also, see that one by one. The C peptide in mammals, including human, insulin synthesized a prohormone, which contain an extra stress called C peptide. Extra stretch. It is the external extension. Okay. This C peptide is not present in the mature insulin. Note it down, my dear. Okay. Just like a DNA already we stored, sticky end, look like that one. C peptide means nothing but the protein molecule. So insulin is also one of the polypeptide protein molecule. It contains 143, I think 143 or 169. Exactly, I do not know. So I will tell you how it will be. How many peptide bond is there? How many amino acids are united together and form as the insulin component? Okay, it's a hormone, but at the same time, it's made up of protein molecule. Some of the hormones are made up of what is known as steroid, fatty acid derivatives. Here, the insulin is a C peptide, they told. So insulin is also one of the protein molecule. Don't forget. Protein molecules means it's connected with the more than one amino acid united together with the help of the peptide bond only. Just note it down. That point is very, very important. Here, what they are telling, no, the C peptide is not present in the mature insulin, and it is removed during the maturation into insulin. So that's why it's called as prohormone. When it undergoes the maturation process, what happened, you know, the last is sticky end, otherwise called as the C peptide stretches, isn't it? Additional components completely removed. That's why that component is called as what is that? Mature insulin. Suppose if it is C peptide present along with the insulin molecule, that is considered as a immature, otherwise called as prohormone. Okay, don't forget, my dear. This is also a very, very important one. C peptide. What is the meaning of C peptide? Extra stretch. Okay, it contains insulin. Contain the extra stretch is called as what is that? C peptide. Suppose if it is presence of C peptide, that hormone is called as prohormone. Okay. When it undergoes the maturation process, the C peptide is removed. So that process is called as prohormone to mature hormone. Okay, prohormone to hormone, whatever it may be. You can think like that. You can imagine. Next one is genetically engineered insulins. So simply called as, what is meant by humulin? Humulin. Any idea, my dears? Ajay? What is meant by
No,
humulin?
sir. No, sir.
What about Dhanalakshmi? Do you know that one?
No, sir.
Okay. Humulin is nothing but the insulin is artificially synthesized in where, you know, the lab. Okay. That insulin structure similar to the, that of the human. That's why they named as a humulin. Human insulin. Okay. Human insulin is also isolated or artificially synthesized in the lab. Okay. So such type of component is called as, such type of product is called as what is that, you know, humulin. It's also one of the process with the help of the E. coli. Okay, it's a vector. Okay, anyhow, just see here, the genetically engineered insulins. Genetically engineered means genetically, what are the technique here, you know about that. First of all, is that identify the insulin secreting nucleotide. Okay, you have to identify particular gene and also the isolate from the insulin. encoded nucleotide from a particular plasmid. Okay, see, I will show you. So then only it will be better understanding for your better understanding. Okay, see this one. This is considered a bacteria. Okay, this is considered a bacteria A. Okay, another one is bacteria B. Okay, this one I will give another structure. Okay, inside there is an extra chromosomal body. Is that the extra chromosomal body is what is that, my dear? The extra chromosomal body present inside. What is that name? This is the extra chromosomal body present in it.
So plasmid.
Very good, my dear. So both are considered as a plasmids. Okay, plasmids. Okay, so this is a considered both are plasmids. Okay, now I isolate this one. Okay, after rupturing this one, what is the cell wall of the bacteria? We can obtain the molecules. So same one is also here. Okay, so this one is considered as a bacteria gene, otherwise called as plasmid. This one is called as B bacteria plasmids. The particular gene is essential for the production of what is that, in this part. Okay, the particular gene. Okay, in this portion. Okay, take an example here, 100 to 120, like that you can imagine. 100 to, this one is a 100 gene or nucleotide. This one is called as 125. Okay, this in between this nucleotide. Okay, in between this nucleotide. Okay, this is the one of the parts. So same here also there, but it is not responsible for the production of insulin synthesis. This gene is responsible for the insulin synthesis. Insulin synthesizing gene. Okay, so you have to use the molecular scissor. Okay, molecular scissor means particularly in our body you have to made a cutting here, restriction enzyme, with the help of the restriction enzyme. So this gene is completely removed. So how it will be? The molecule will be like this, isn't it? So we made a cut. Okay, so we made a cut, and also see that one. Remaining portion will be like this, isn't it? Remaining portion will be like this. After cutting, this is the responsible gene of the human insulin and insulin secretion process. The other genes, okay, this one. Likewise, here also I have to make a cut from this portion, completely removed. So that is called as deletion process. So the B look like this one, isn't it? B look like this one. So you can imagine. So the A, the responsive gene also replaced to here to B. Okay, I can indicate the different colors because it's the same, that's why I can use it a different colors. After the recombination. So this is the, what is that? A gene. Okay, so the A gene responsible for what is that? A gene responsible for the production of what is that? Insulin. Okay, insulin. Okay, insulin. Okay, so here what are the process that occurs? You have to make a cut from the A gene, and so use the restriction endonucleases and remove it and replace to the B genes. Okay, this is the B one. Okay, this is the B gene, this is the A one. Okay, so responsible gene, it's also replaced here. After that, what happened? It may be grow into a E. coli bacteria. Take an example. So another bacteria like this. Okay, it is nothing but the vector. Okay, so it's a vector. So inside we are going to introduce. This is considered a new one. That's why I have changed the colors. So this one is a new one. After the combination of A and B genes. After the combination of A and B genes. Okay, so this is the important. So yeah. So both the complement genes. So this is called as a human insulin producing genes. It now change into A plasmid to P plasmids. Then this is the recombinant technology with the help of the enzymes. One is called as restriction enzyme. So when we are adding the B gene, this is the A gene, isn't it? A gene is all B genes also united together with the help of the enzyme is called as ligase enzyme, isn't it? Ligase. The cutting is also made, and also linking also done in both the cases. Ligase enzyme, this process. After the finishing of ligases, we are introducing into the E. coli bacteria. E. coli is act as a vector. E. coli is act as a vector. Inside, what happened, both the gene present in it, both the genes are what happened, undergo the replication process. You will get it the human producing insulin inside the E. coli bacteria. From this one, we can isolate the which one, insulin secreting genes. That insulin secreting component, they can produce more insulin in the commercial. These are all occurs in where, laboratory work, in lab oriented. Highly sophisticated lab is also there, okay, and also commercially oriented. Nowadays we are using humulin for those who are all suffering from the diabetes patients. That patients are using the injection, isn't it? That injection is also commercially prepared. It is also one of the process. And one more is also there. One more is there. Take an example of pig. Okay, the pig. Okay, so the pig insulin is also similar to that of the human being. Human being. It is also similar to human. Okay, so this is also a human being. How we know the peptide molecule is that this is considered a peptide molecule. Okay, this peptide molecule contains a particular nucleotide sequences also there. Here you have to make small changes. We have to make a small changes, physiological changes, addition, deletion, whatever may particular the sequences. The particular sequences will be removed from this one, or add up some other component. Okay, addition of some other components. So that time what happened? The pig insulin compound acts as a human insulin. When there is some physiological changes occurs, okay, on the pig insulin compound. Okay, so finally what happened? The pig insulin compound is also converted into human insulin, and also it is also used for the commercial purpose. So this is also another technology. This is also another technique. This is the normally genetically engineered technique. What is the meaning of genetic engineering means? What is that meaning? We can construct our own DNA sequences. Otherwise, we can construct our own plasmids also. Here we are doing the own plasmid. That is very, very important. This air quality will be isolated, identified and isolated from the particular plasmid, and also introduced to the another one. Okay, and also linked with this one. So both are linked. After that, the growth. The growth means what is that? You can introduce inside the E. coli. As a result, what happened? Undergo the replication process within the course. What happened? It will produce both the genes A and B gene. Likewise, the resistant genes also produced tetracycline resistance and ampicillin resistant and penicillin resistant. Like that, we can identify the gene. Which gene is it? For example, this one is A. This is called as ampicillin. This one is what is that? P. This is penicillin resistant, and another one is T. So this is tetracycline. Okay, response resistant genes. Like that, we have to identify. After the identifications, it may introduce into the same technology. So as a result, what happens? Suppose if you are introducing a, you will get ampicillin resistant gene production here in this part. Suppose if it is penicillin resistance, you will get the penicillin resistance here. Okay, tetracyclines are also tested. Sometimes they may use more than two tetracycline, the ampicillins also both. Likewise, the technology is what is that used for our human life. Okay, my dear. Understand, my dear? Ajay?
Yes, sir.
Okay.
Yes,
What?
sir.
Okay. Okay.
Yeah.
Okay. See that? Sorry. Okay. See, next. Yes. Genetically engineering insulins. So here, just imagine The technology is called a genetically engineered. Genetically, gene we are using our own style engineer. We can make our own style. Okay, engineer. Insulin means but one of the hypoglycemic effect hormones. The human insulin was prepared by the recombinant DNA technology. This is already recombinant with two genes united together. Okay, this insulin has a two short polypeptide chain. The insulin, it has a two polypeptide chain. Already I told you, A and B polypeptide chain, and also it has some molecular amino acids, number of molecular amino acids also. Sometimes they may ask the question. I will tell you next day. A and B, which are linked by the disulfide bridge. That is also the combination. Suppose the A and B chains are united together with the help of the which bridge? Disulfide bridge. Don't forget, my dear, this is also one of the NEET question. Disulfide bridge. A and B. Okay, it's nothing but the two polypeptide chains, the short polypeptide chain, united together with the help of disulfide bridge. Okay, don't forget. So I think that's all about the basic introduction, my dear. Today we will start into the what is meant by introduction, who discovered, how it will be essential for the human as well as the biological aspects, how it will be used. We will discuss. First of all, is it know what is meant by biotechnology, how it will be used. Okay, introduction part. Now only we are entering into the introduction part. Before that, already we saw it. What are the technical terms? What are the other terms also comes inside the lessons. The plant and animals and microbes have been used by the human for nutrients and the development of the product for consumption, such as beer or bread. These also one of the, what is that? Consumes human purpose only, commercial purpose only. Okay, man-made process, whatever it may be. It is also used for the technological aspects. Such a technology is also introduced here. See here, the technology advancement has allowed us to exploit plant, animal, and microbial. Suppose the highly resistant plant, highly resistant animal, highly resistant microbial. It's also very, very important. Take an example. Nowadays, there is a corona is also, once again, it's repeated, isn't it? How you have to take care. Same process also repeated again and again, isn't it? You have to use the mask and high immunity powers and hygienic. You have to maintain within your body as well as outside or inside, both. This is also very, very important. Otherwise, what happening? It will lead to death. Technology advancement has allowed us to exploit plant and animal microbial wealth to provide product of commercial or pharmaceutical importance. That is very, very important. Some of the microorganisms are used as a, what is the medicinal purpose. And already about streptomycin. Streptomycin is also isolated from the Streptococcus bacillus, isn't it? Streptococcus, one of the microorganisms. Take an example of penicillium. Penicillium is also isolated from the one of the fungi is called as Penicillium notatum, Penicillium chrysogenum, isn't it? Both are plant. Okay, plant and animals. And I already told you, the pig insulin is also used as a commercial purpose. Plasmids, plant as well as elements, we can isolate it, different sources. Both are entirely different, isn't it? Plant sources, animal sources. But similarity, how it will be? The insulin is similar, how it will be. Okay. And one more is that, take an examples of the jellyfish. Jellyfish as well as the tobacco plant, both are combined together. You will get the new variety. Okay. Just like a crashing of the hybridization takes place with the within the two animals, lion and tiger, lion and something else. Just to go and check it, new genetically modified, what is that? Animals, just to go and check it in the net. You will get 6 animals, different aspects. Okay, just to go and check it tomorrow. Okay, right here. Commercial are the pharmaceutical importance. All these activities, research and development, research only, research. What is the meaning of research? Whether it may be beneficial or non-beneficial. How will you identify it? Take an example, COVID, co-vaccines, vaccines also isolated by the technique, genetic engineering, biotechnology. Development, how it will be developed. This one is used for the primitive stage, and someone is and also used for the booster, how it will be improved. Okay, which part we can use as a medicine, that things also. You know about the cinchona, okay, colchicine. It is also used for the malarial drugs. One of the bark of the cinchona. Bark means already in our body, it's the outer layer of the plant. All this activity, research and development fall under the big umbrella of biotechnology, whatever it may be. Whatever, it's a very, very interesting interesting subject, biotechnology. Very interesting. Okay, you have to do gene. You do not know how to, whether the cell is present. Okay, inside the cell there is a chromosome is there. We can isolate the chromosome. We can visually, what is that? We can see it our own chromosome structures. What are the pattern is there? Okay, then how it will be changed? Okay, that things also, how it will be isolated. There is a technique is there. Even take an example, electrophoresis, DNA fingerprinting. So we can use some advanced methodology. Okay, see that one. That is comes under the one umbrella, isn't it? Under the big umbrella of biotechnology. The term biotechnology was first coined by Carl Erik, a Hungarian engineer in 1919. Don't forget, my dear, this is very, very important. Biotechnology, the first introduced by the name of the scientist called as, what is it? Carl Erik. Hungarian engineer. Sometimes they may ask the question, which country? Sometimes this may be which year. Okay, this is a match the following or character questions. Sometimes they ask the questions. Don't forget, my dear, this is very, very important. Okay, go to the next one. Naturally occurring animals and plants or microbial strains have a few limitations. For them to be utilized for desired product due to the following reasons. Why we are using this one? Natural animals and plants also having a particular limitation having, isn't it? Then how it will be used as a commercial purpose or genetically engineering, how it will be used. Okay, see that. Production of undesired product. Undesired product. Suppose if it is not possible, like that they were told, we can use it. And how it will be this year, how it will be used as a useful product. That is why production of undesired product, the main one under the biotechnology. Secretion of toxic metabolic by the product. Sometimes what happens, it may give a positive effect. Sometimes it may be negative effect. Some of the substances, Bacillus thuringiensis, ground gall effect. Okay. Ground gall effect, toxic substances also produced from the BT cotton, isn't it? So the secretion of toxic metabolites by the product. This is also another important one. Inability to withstand harsh biochemical process treatment is also. Inability to withstand harsh biochemical. That means some of the animals or plants, when you are applying the chemical substances, what happens? It may fight resistant to the chemical substances. Such type of animals or plants, what are the genes responsible for the resistant power or treatment? That genes also we can easily identify it. That is also very, very important, my dear. Okay. And higher production cost. Suppose any one of the plant or animals, it make high productions. How it will be reduced? That is also one of the another technology they were used with the help of the biotechnology. And then susceptible to disease and other environmental conditions. Already you know, but some of the plant they can resistant to the cold or tropical region, high temperature or moderate, whatever it may be. Some of the plant also like that. Some of the animals also like that. Okay. Not only that, and also some of them are susceptible to the disease and other environmental conditions. Disease means, some of the plant it affected. Take an example, Edward Jenner, one of the scientists, isn't it? He will not affected by the cow pox. I mean, smallpox, chicken pox. Why? Because the cow excretes whether urine or the cow dung. It has the antibacterial capability. So that person, it will not affected by the particular diseases because some of them are some microorganisms. That's why we are using cow dung. In ancient ages, the cow dung used as a, what is that? They are sprinkling in front of his house. The antibody, it's also one of the antibiotics. Nowadays, they were used as some powders. Okay. Susceptible to the disease and the environmental conditions. And then the European Federation of the Biotechnology has defined biotechnology as European Federation of the Biotechnology. This is also simply called as EFB. He has given us a particular definitions, okay, of the biotechnology. The integration of the natural science and organism cells, parts thereof, and molecular analogs for product and service. Okay, what is that? The integration of the natural science and the organism cells part thereof, and molecular analogs for the product and services. This has given by the EFB. Okay, that is nothing but the European Federation of Biotechnology. Okay, see that one. So remaining things will we see the next classes, my dear. So before that, we summarize all those things. What are the things already we discussed, you know about? So one is called as what is that? Polymerase chain reactions. Here what happened, you know, DNA or DNA. So recombinant DNA we will obtain, how it will be. What is my restriction enzymes? Okay. What is my stem cells? Okay. What is my stem cells? And sticky end. What is the meaning of sticky end? It is a extended of the double stranded molecule. There is a single strand of molecules also present in the extended one, complementary gene. That extended gene is called a sticky ends. Taq polymerase, it's nothing but one of the heat stable DNA polymerase isolated from the thermophilic vector, that is called as thermus aquaticus, used in the PCR. This is very, very important medium. And TI plasmid, otherwise called as what is a tumor inducing plasmids, an extra chromosomal double stranded and self-replicated DNA molecule found in the Agrobacterium tumefaciens that cause ground diseases in plant. Don't forget ground diseases, ground gall diseases caused by the TI plasmid. This is very, very important, my dear students. Agrobacterium tumefaciens. And then tools of recombinant DNA technology. This is also very, very important. Restriction enzyme, it's also you know about molecular scissor. Polymerase, it's also recombinant enzymes. And ligases, the linking of the two DNA molecules, vectors, plasmid, cosmids, phage, bacteriophage, bacteria, and host organisms. Okay, this may be plant or animal. Simply, if you want to mention it specifically, means E. coli is the main one. Okay, and ADA, so the otherwise called as adenosine deaminase. It's also very, very important for the immune system to functions. This disorder is caused due to the deletion of the genes for the adenosine deaminase enzymes. Okay, due to some lack of immunity. Bt cotton is also very, very important. It is also transgenic plant Bt toxin. Genes were isolated from the Bacillus thuringiensis and were incorporated into the cotton plant. Okay, this is also very, very important. Another one. How it will be act as, how it will be used, we will see in the lesson. Okay, cryogene. Cryogene is also one of the gene is also isolated from the where in the Bt toxins. It encoded or coded by a gene named by cryogene. The gene is produced a cry protein. From where you know Bacillus thuringiensis. It's called as cry protein. Okay. And then C peptide. C peptide, you just note it down, insulin must mind it. Okay. In this category, there is a pro-hormone. What is the meaning of pro-hormone? It's nothing but the extra stretches present in the hormone. That hormone is called as pro-hormone. Suppose if it is matured, what happened to the extra stretch? Completely removed. Okay. So that is what genetically engineered insulin. What is the meaning of genetically engineering? It's also used for genetically prepared or commercially prepared. Okay, in the laboratory aspect, technology aspect, DNA technology aspect, genetic engineering aspects. Okay, such gene is called as, such enzyme is, such hormone is called as genetically engineered insulin. That is called as humulin. This insulin has a two sort polypeptides, and don't forget, my dear, A and B. But both are polypeptide with the help of which one in disulfide bridge only it will be united. Otherwise, it will be what happening? It will not be strengthened. Okay, so we'll finish it up to this level, my dear students. We will see the remaining things on the next class. Thank you so much. Bye. Take care, Ajay. Good night.
Thank you, sir.
Thank you, ma'am.
Thank you, sir.
Thank you, ma'am. Okay.